Good, very good. So welcome to this masterclass. In this masterclass, we're looking at AS Biology for the Cambridge International exams. And this paper is from the summer season 2015. It's a multiple choice paper, paper 11. And what we're going to do during this masterclass is rather than go through every single question, the mark scheme is available, you can look up quite a lot of this, is look at the report that the examiner made after the students had taken the exam. And the examiner identified between 10 and 15 questions where students particularly had difficulty. So rather trying to do an overview of all the questions, what we're going to do in this masterclass is do a deep dive into the questions that the examiner identified that students had the most difficulty with. And for the rest of the questions, they're fairly straightforward. They can be looked up easily in the mark scheme and in revision notes or textbooks. Okay, very good. So the examiner actually came in at this question, number 3, and the comment from the examiner was that many candidates found this question difficult and could not manipulate the information in the question satisfactorily. And so what you've got here is the view that you see when you look down the eyepiece of the microscope. And what's happened is that a tiny ruler has been placed on the stage of the microscope. That's the thick black lines that you can see at the back. And then in the eyepiece, there's a little scale called a graticule, which is the numbers 0 to 100 that you can see. And so this is the this bit here is the part that's on the microscope stage. And this piece here is the graticule in the eyepiece of the microscope. Okay, so what you've got here is this is a ruler, and it says that this division here is 0.1 millimeters. So that's the actual size of an object on the microscope stage, because they've placed this tiny little ruler on the microscope stage, and it's now magnified, and the image size is then the image size that's measured with this graticule in the eyepiece. And if you see there, we're going from 50 to 90, so there's actually 40 millimeters along that graticule scale, and that is the image size. So you've got the actual size of something. In this case, that distance there is 0.1 millimeters, and the image size that you see is being measured by the graticule in the eyepiece at 40 millimeters. And so you need to know that the magnification then is the image size, which was, what was it, 40 millimeters, divided by the actual size. Now this little division here on the microscope stage, that division is 0.1 millimeters. And if you divide 40 by 0.1, then you get a magnification of 400. All right, so that's, we've worked out from this first view of the microscope, the magnification. Now, once the magnification has been determined, we swap out what was on the microscope stage, these divisions here, and instead put on the microscope stage the sample of interest. You can see that it's here. And we want to measure the width of the chloroplast. You can see the plants are here, the chloroplasts. And, well, they're all pretty uniform in size. But if you measure the width of the chloroplast, well, against this scale here, yeah, you can measure there and there, can't you? You end up with a width of about 4 millimeters. So this is now the image size, isn't it? Because we've measured against this graticule in the eyepiece. And so to get from the image size actually to the actual size, then again, you've got to use your equation that the actual size is equal to the image size divided by the magnification. Now, the magnification, if you remember from last time, was 400. And the image size here is 4, isn't it? So that means we've got to divide the 4 millimeters that we measured on the graticule by 400. And that gives us a result of 0.01 millimeters. You've just divided 4 by 400, and that's 100th of a millimeter, 0.01 millimeters. But you've got to choose one of these answers here, and you've got to remember your metric scale. So millimeters is 10 to the minus 3 meters, isn't it? Micrometers is 10 to the minus 6, and nanometers is times 10 to the minus 9 meters. Now we've got a value here of 0.01 millimeters, and that's the same as 10 micrometers, because there's 1,000 micrometers in 1 millimeter. And so we've worked out then that the size of the chloroplast was 10 micrometers. And really the trick there was to understand that what you see on the microscope stage, well, that is the actual size, and what you're measuring with the graticule is the image size. And then you've got to use your relationship between actual size and image size to give yourself the magnification. And then once you know your magnification, then you can work out the actual size, in this case, of the chloroplast that you saw on the microscope slide. Good. Okay, so the next question actually was this one, and the comment from the examiner, strangely enough, was that the majority, more than 50% of the candidates, did not know what a typical eukaryotic cell contains, which is kind of surprising, really, because the differences between eukaryotic cells and prokaryotic cells, you'd think, would be fairly well established in students' minds. Well, the thing here, you can look at these answers here, and well, in a eukaryotic cell, that's going to have a nucleus, isn't it? Well, there's going to be linear DNA in the chromosomes inside the nucleus. So that's a definite case for eukaryotic cells. These ribosomes, these site of protein synthesis, well, you've got the smaller one here and the larger one here. The larger one is bound to the rough endoplasmic rectilium, and the smaller one you can find in some of the organelles, such as chloroplasts or mitochondria. And these are organelles, aren't they, that you find in the larger, more complex eukaryotic cells. So that's going to be found in a eukaryotic cell because it's found in organelles. That's found in a eukaryotic cell. The chromosomes, of course, are part of the nucleus in a eukaryotic cell. And this was the one that I think threw students because the circular DNA is found, isn't it, inside mitochondria. Yeah? So the circular DNA is inside mitochondria, and the mitochondria are themselves organelles within a eukaryotic cell. So that is also found in a eukaryotic cell, but if you like, it's buried one layer deeper, so to speak, within the organelle within the eukaryotic cell. And so for this answer here, 1, 2, 3, and 4, all of those were found within the eukaryotic cell? And answer A was the right answer there. Okay, so moving along, the next question that the examiner flagged up for students was question number 10, actually. Where's 10? Okay, so there's a whole question here doing a comparison or a contrast between amylose and cellulose. All right, so we'll just revisit those two polysaccharides for a moment. So before we dive into the table here, so both these are polysaccharides that are found in plants, aren't they? Because cellulose is a polysaccharide that's used in the cell wall, plant cells, and amylose is one of the polysaccharides that you find in starch, which is an energy store for plant cells. So these are both polysaccharides found in plants, and we have to look at the similarities and differences between them. Just to take amylose, that's one of the polysaccharides in starch. The other one is amylopectin. And the amylose is a polymer of alpha glucose monomers. So you just need to know that the alpha glucose looks like this, doesn't it? There's an oxygen atom there. And you can see here, if I draw the carbon in there, if you count round 1, 2, 3, 4, 5, 6, you've got 6 carbons. So this is a 6-carbon sugar. It's a hexose. And when you have alpha glucose, the alcohol group there, the OH group, is pointing downwards like that. And the other groups, as you go around the glucose molecule, that's on carbon number 2, that's on carbon number 3, this is carbon number 4 over here. And then up here, on carbon number 6, you've got another OH group, another alcohol group as well. So counting round, this was carbon 1, 2, 3, 4, 5, and carbon 6. And because the OH group is pointing down there in that direction, this is now alpha glucose. And the polymer chain forms because you get another one of these molecules coming alongside it here. I'll just draw it in. Hexose sugar. You've got carbon 6 up there. You've got the alpha glucose here, haven't you? And then you've got, that's number 1, number 2, number 3, and this one is number 4, isn't it? Right, so you've got 2 alpha glucose monomers, and they're going to combine together. It's called an elimination reaction, because you're losing, aren't you, a water molecule. So you're taking H2O out, you're eliminating H2O. It's an elimination reaction or a condensation reaction. And you end up with a bond that's formed here. If I just draw it in, which is the glycosidic bond, because you've joined 2 of these glucose units together. And you can see if we count round, we had the bond there to number 1 of this first monomer. And if we count round here, 1, 2, 3, 4, you've got a bond between number 1 carbon through to the number 1 carbon in this first alpha glucose monomer, and number 4 carbon here in this second alpha glucose monomer. So when you just, and that pattern would continue because you're building up a large unbranched chain of this amylose polymer. And so you're looking at a 1 to 4, we hit it there, glycosidic link. And all these glucose monomers are all lined up, yes? So that you could say there's no rotation between each monomer in the chain. So that's amylose. Now, if we want to look down here, let's see, amylose, alpha glucose, yes, alpha glucose. Well, this one talks about a rotation and 1,4,1,6 linkages. Well, that's wrong, isn't it? Now this one, alpha glucose, no rotation, 1,4 linkages. That looks good. Alpha glucose, no rotation, 1,4 linkages. That looks good. Beta glucose, no rotation, 1,4 linkages. Well, that's wrong, isn't it? So just by process of elimination, after considering amylose, then we've got down to either B or C is the answer. So that's amylose. Let's look at cellulose now. So with cellulose, again, you've got 1,4 linkages. So let's draw our glucose molecules again. Okay, we're going to have 2 of these, aren't we? But this time, if we draw them, you can see that actually one of them is flipped compared to the other. It's rotated through 180 degrees. And so our carbon number 6 has now ended up down here, isn't it? And then you've got these other OH groups around the glucose molecule. And this one is in a little bit more familiar orientation. It's a bit like the ones we drew up here. Here's your number 6 carbon. 5 is there. 4 is there. 3, 2. And when you come to 1, now look at the difference here. Now that OH group is above the plane. You see it's above, it's on the same orientation as this carbon 6 over here. So that's no longer alpha glucose, but that's beta glucose, isn't it? Because you've got the OH group pointing upwards here, but you had it pointing downwards over here in the alpha glucose. So this is beta glucose. And you can see here we're about to form one of these elimination reactions or a condensation reaction by eliminating a water molecule here. So let's eliminate that. And we formed a glycosidic bond there. Now, if we count round, that's 1, 2, 3, 4, 5, 6. And if you count round on the other way, then this again, this is actually, this is carbon number 1, isn't it, here? All right. So you've got, again, a 1,4 glycosidic bond, or glycosidic link, but you've got beta glucose, and this molecule is in the same orientation as we had the ones up here, but this one's been flipped round, hasn't it, by 180 degrees. And so the next one in the chain here would be flipped around 180 degrees, and then the next one another 180 degrees, and so on. And so you've got here, for cellulose, 180 degree rotation between each monomer in the polymer chain. So let's go over here and see what we've got. Beta glucose, yes, no rotation, that's not right. Beta glucose, 180 degrees rotation, 1,4 linkages, that's good. Alpha glucose, that's wrong. Alpha glucose, that's wrong. And so this line here, B, is the correct answer. And the examiner said that a lot of candidates couldn't really work out or didn't really understand these structures as we've drawn them out here of amylose and cellulose. And very, very many students had the misconception that cellulose, this one, contained alpha glucose. It doesn't. Cellulose is made of these beta glucose monomers. So the examiner is telling you here that you need to learn these hexoses, these 6-membered rings for glucose, which one is alpha and which one is beta glucose. You need to learn the numbering around the ring, and you need to know that in these polymers, sometimes the monomers just join up like this, but sometimes every other monomer is flipped 180 degrees. There's 180 degrees rotation. And that level of understanding of how these biological polysaccharides are built up from the individual hexose units was very poorly understood by the students. So that's worth spending some time learning that. Okay, 11. Next one that the examiner identified that students had a lot of difficulties. And again, we should just work through understanding the structure of collagen. All right. So in collagen, the first level of structure you need to understand is that you have a polypeptide chain. So that means a series of amino acids all joined together through peptide bonds. And these polypeptide chains in collagen, they're about 1,000 amino acids long, or you might say 1,000 residues long. So these polypeptide chains, and they form a very loose kind of helix like this. And that helix is kept loose because every third of these amino acids, every third one is glycine. Now, if you look up the structure of glycine, as an amino acid, its side chain is kind of not a side chain at all. It's just a hydrogen atom. And so when you have every third amino acid in the chain is an amino acid with no side chain, it means that you don't get a very compact or pulled together helix forming. And what happens in glycine is, if I can just get some other colors here, is you get 3 of these polypeptide chains all close together. And they're held together, those 3 polypeptide chains, by hydrogen bonding. Like that. All right, so that first level of detail is you've got 3 polypeptide chains, which have a kind of very loose structure here, because every third amino acid is glycine, and they're held together by hydrogen bonding, and that's called a triple helix. And you might say then that one molecule of collagen is a triple helix of these 3 polypeptide chains. Now, once you've got that triple helix built, you can then start to line up many triple helixes or helices next to each other. So if I use the colors here, you'd have that one. I'm just drawing them like this now. So that's one triple helix. And then you've got another one like this. And then you've got another one, and so on. I could keep drawing, but I'm just going to draw, well, let's draw 4 so you don't get mixed up, because we could just keep going on like this forever. Okay, so each one of those is this triple helix, isn't it? But when you have many triple helixes together, the whole thing is called a collagen fiber. And these collagen fibers are held together because these triple helices are joined to each other through crosslinks here. And those crosslinks are covalent bonds. So that collagen fiber, you see, is made up of many of the triple helices joined together with covalent bonds. And each triple helice, or each triple helix, is made from these three polypeptide chains, which are kind of loosely bound together with hydrogen bonding. So you've got to look quite carefully at these questions here. The actual answer is this one here, it's A, but let's just look at the distractors for a moment. So if you look at this one here, it says a group of 3 collagen fibers forms structure, which is a kind of helix. Well, that's not true, is it? Because it's a group of 3 polypeptide chains forms a collagen molecule. So they're kind of playing around with the vocabulary here, but that's wrong, isn't it? It's not that you're getting 3 collagen fibers, you're getting 3 polypeptide chains forming a collagen molecule, and then many collagen molecules are joined together to form a collagen fiber. So that's a kind of a distractor that I think is quite easy to choose incorrectly. Now each of the collagen polypeptides, so far so good, in a collagen molecule, good, has a regular spiral arrangement of many alpha helices. Well, that's close, but these are very loose helices here. And they're loose because every 3rd amino acid is glycine. So they're not typical alpha helices, they're much looser than that. So that's the distractor there, that's wrong. And it says here that peptide bonds are present between the amino acids of the same polypeptide and between different polypeptides in the collagen molecule. Well, can you remember here that these polypeptide chains are being held together by hydrogen bonding? It's only when you put the many triple helices together in a collagen fiber do you join the individual triple helices with these crosslinks to form the collagen fiber. So these peptide bonds are present in the collagen fiber, not in the collagen molecule. So this is all quite sort of accurate work, really. You've got to know about the structure of collagen quite accurately to avoid getting drawn into the trap of these distractor answers here. And so the only one that's left, if we positively select it, is it's the collagen molecule. Here it is. That's the collagen molecule. Has a high proportion of the amino acid glycine, which is true, because each one of these chains, every 3rd amino acid is glycine, and it says which has a very small side chain. Well, it's the smallest possible side chain you could get in glycine. It's just a hydrogen atom. So we can negatively exclude those distractors. They're quite easy to get, but positively identify that as the answer that we needed. And the examiner said for question 11 that many candidates didn't understand that the peptide bonds were in between the triple helices in the collagen fiber, and they wrongly thought that peptide bonds were involved in keeping these 3 polypeptide chains in the triple helix. They're not. This hydrogen bonding is being used to hold those 3 polypeptide chains together in the triple helix. Okay, good. So next question that the examiner flagged up for students was this one here. Maybe I can just try and make that a bit smaller so we get everything on one page. I won't do that. Can we make that a bit smaller? No. Oh, there we go. Okay, good. All right. So this is a question about enzyme catalysis, and we're looking at a competitive inhibitor. All right. Now, let's just start off then just thinking about the enzyme itself and the substrate concentration. So if you remember, an enzyme has an active site which has a very specific shape. And that specific shape is matched or is complementary to the shape of its substrate. So they're going to fit together. It's a so-called lock and key model, isn't it, for putting the substrate exactly in to the shape of the active site. And what's happening is you're adding more and more and more substrate, and eventually you add so much substrate that all these active sites are completely saturated and no matter how much substrate you put in, you're just adding substrate to the queue, and you can't get a greater rate of reaction because already the active sites are completely saturated with substrate, and no matter how much more you put in, you can't speed the rate of reaction up any faster than it already is. And that's why you get this increasing substrate concentration, and you get this rate of reaction here tending to a constant. And you know that that is the maximum rate of reaction of a Vmax. Now, a competitive inhibitor is like a substrate. It has a similar shape because it can compete, but it's not a substrate. Okay? So there's going to be a competition set up between the substrate and the competitive inhibitor. Now, at a low concentration of inhibitor, these two are going to be competing for the active site. And so the inhibitor will reduce the rate of reaction. But as you increase the substrate concentration, the competitive behavior of the inhibitor is going to become less and less dominant, because there's going to be so many substrate molecules that the few inhibitor molecules have little effect. And eventually you're putting in so much substrate that the inhibitor is having hardly any effect at all, and so you actually tend to Vmax again. And so this is looking like the expected graph here, because that's the graph for the substrate on its own, tending towards Vmax. Now, when you put the competitive inhibitor in, initially at low concentrations of substrate, it reduces the rate of reaction. But as you increase the substrate concentration, the inhibitor has less and less impact, because it's getting crowded out, so to speak, by the increasing concentration of the substrate. And so as you increase the substrate concentration, well, eventually you tend towards Vmax again, because the inhibitor is crowded out by the substrate concentration. And that same argument applies for a higher concentration of competitive inhibitor. And you can see that all these three lines, if we continued them, are all trending, aren't they, to Vmax, where at that point the substrate concentration is so high that it's outcompeted all the inhibitors and saturated the number of active sites. And so that graph there is the correct answer. And the examiner said for question 14 that only about half the students were able to work this question correctly by working with ideas of Vmax, saturating the active sites of the enzyme, and the competitive behavior of the inhibitor, and how eventually it's crowded out by the increasing substrate concentration, such that the competitive inhibitor has minimal effect, and the graph trends towards Vmax again. Good. Okay, so next question up that the examiner identified was question 16. This is talking about passive and active transport. Now, this is a cell. Maybe if we draw the cell, it's a bit easier to see. And this is the cell's surface membrane here. And inside the cell, we have the concentration of sodium ions is low, but the concentration of potassium ions is high. That's inside the cell there. Now, outside the cell, you've got the reverse, where you've got the concentration of sodium ions is high, but the concentration of potassium ions is low. Now, just by diffusion, any substance is going to diffuse from a high to a low concentration. So potassium is going to diffuse, isn't it? So potassium is diffusing that way. Now you've got sodium is at a high concentration outside the cell. Sodium is at a low concentration inside the cell. So sodium is going to be diffusing that way. Okay, now how are we doing for the question down here? So, well, let's keep going actually for the moment. So we did the diffusions. Now, you've got a protein here in the membrane of the cell, which is doing active transport. It's using ATP to pump ions against the concentration gradient. So now, let's take this one here. This is going against the concentration gradient. That must mean that this one is active transport of sodium, because we're pumping sodium from a low to a high, which is against the kind of natural order of things, if you see what I mean. It's against the way diffusion would happen. It's active transport. The cell is using energy to pump the sodium against the concentration gradient from a low to a high concentration. And similarly, this one here, you're pumping potassium from against the concentration gradient from low concentration potassium to high concentration potassium. So that is active transport of potassium. And this one here, this was a diffusion of sodium ions. And this one here was diffusion, excuse my writing, of potassium ions. So 1 is diffusion of sodium ions, 2 is active transport of potassium ions, 3 is active transport of sodium ions, 4 is diffusion of potassium ions. And if we look there, active transport of potassium is number 2, yes. Active transport of sodium is number 3, yes. Diffusion of sodium is 1, yes. Diffusion of potassium is 4, yes. We got it. And the examiner's comment on question 16 said that many candidates actually found this manipulation of information quite difficult. I think really you've got to be, under exam conditions, you've got to take a few deep breaths. You've got to kind of slow down a bit, because it's really easy to get muddled up here. Just work it through calmly and slowly. Label things up if you need to. But actually, the level of understanding is quite easy, I think. It's fairly obvious that diffusion goes from high to a low concentration, and the active transport goes against the concentration gradient from a low to a high concentration. I think that's fairly obvious. So, I think the information here is not difficult to understand, but it's being calm under exam conditions to be able to work through carefully and make sure that you get the right answer. Okay, so next one for the examiner was question 21. Scrolling up here. And in question 21, you had to have a pretty good understanding of the chemical structure of DNA and RNA. Now, the other thing that you've got to watch for here is, if you look in the question, it's talking about some RNA. Now, that's a kind of clue, because, as you know, there's 2 types of RNA. There's messenger RNA, isn't there? And then there's transport RNA. And so when it says some RNA, it's probably meaning, well, we've got to think about the differences between messenger RNA and transport RNA. But let's just have a think a little bit more about the structure of these nucleic acids. Well, for both DNA and RNA, you have to remember that we don't have a hexose sugar like we did earlier on with those plant polysaccharides, but you're now having a 5-membered ring, and so that is a pentose sugar. Yeah? And for DNA, you've got numbering 1, 2, 3, 4. You've got 5. That's the 5 carbon there. And that's 1, 2, 3, 4, 5. And then you've got that used to be an alcohol group in the pentose sugar, but in DNA, you've got a phosphate link, haven't you? Having draw over here. Okay, and that's coming into the next number 5. There's your pentose sugar, 5, 4, 3, and then this is the next phosphate link, and so on to the next carbon 5, the next pentose sugar. I'm rushing now a bit, and that's carbon 3 there. So you can see here that we're going from 5 to 3. There's a direction in these nucleic acids. All right, and on carbon number 1, that's where, if I can rub out that 5 to 3 direction, that's where the nitrogenous base goes. Remember, there's 4 different bases in DNA and RNA. And so in DNA, you've got one strand. This strand was going 5 to 3, wasn't it? And there's another strand which is going the other way. It's anti-parallel, going 3 to 5. So it's going up there, and that will have its own nitrogenous bases, and they'll be forming hydrogen bonds between the 2 strands. And so DNA is double-stranded nucleic acid, and those 2 strands form a double helix. And you have, for DNA, you're having adenosine, and that's forming 2 hydrogen bonds to thymine, and then you've got guanine, and that's forming 3 hydrogen bonds to cytosine. So that's for DNA, and you get something similar, but not quite the same in RNA, where you're getting same guanine bonding to cytosine there, triple bonds, or triple hydrogen bonds. But in RNA, you've got the adenine, but it's forming 2 hydrogen bonds to uracil. So that's the difference between DNA and RNA. The other difference, of course, is that D stands for deoxy, because there's no hydroxide group, OH group there, whereas RNA has hydroxide groups on all of the carbons in the pentose sugar. So it's ribonucleic acid, not deoxyribonucleic nucleic acid. Okay, so that's DNA. It's double-stranded. Now RNA is single-stranded. Okay, so it's just a single-stranded of these pentose sugars joined to a phosphate group like this. And the thing you've got to watch here is that RNA is single-stranded, and you've got your messenger RNA, and you've got transport RNA. And the trick for this question is that the transport RNA is the single strand. There it is. But transport RNA is folded, and the typical kind of drawing for that is to think of the transport RNA as having a so-called cloverleaf shape. So you get something like a fold here, then a fold here, a fold here. And that's all single-stranded, but we're folding it. And the transport RNA, its function, isn't it, is to bring in an amino acid to the site of protein synthesis. So that's the amino acid there. Not important, particularly for the question we're talking about. But the transport RNA, even though it's single-stranded nucleic acid, is folded. And those folds are held together by the hydrogen bonds between the nitrogenous bases that form part of the nucleic acid. So this is where this answer comes in, because hydrogen bonds form between bases in some RNA, while in transport RNA, because transport RNA is single-stranded nucleic acid but folded, and those folds are held together by hydrogen bonds, and those hydrogen bonds are between the nitrogenous bases. Well, it's true that hydrogen bonds form between bases in transport RNA. So which row correctly identifies features of RNA and DNA molecules? Well, we know that's true, and that's true. If we look at the other comments, DNA and RNA both contain purine and pyrimidine bases. Well, that's true, isn't it? Because these bases up here are either derived from purine or derived from pyrimidine. So that's right. And our last comment here, DNA and RNA both contain a pentose sugar. Well, that's true because these nucleic acids don't have the 6 carbon sugar like we saw with alpha and beta glucose earlier on. We're now looking at a 5 carbon sugar, a pentose sugar, and it's true that both RNA and DNA have this 5 carbon sugar, the pentose sugar. So that, and that. And so if we look across the table now, the only row that has got ticks in all columns is A. And the examiner's comment on that for the students was that just only about 50% of the students knew this feature of transport RNA, that it is single-stranded, yes, that it's RNA, yes, but it folds, and those folds are held together because hydrogen bonding occurs in the folds between regions of complementary bases. So that's just a bit of accurate understanding about the structure of transport RNA that about half the students were missing, according to the examiner. Good. Okay, so after 21, the next one that the examiner flagged up was 26. Okay, this is a thin cross-section through a vascular bundle in a plant. And students needed to know the structure and the function of the different types of cells that you get in a vascular bundle. Well, these ones here, those ones are the xylem. Okay, and coming up through those are just through sort of mass transport of water is water with dissolved ions inside of it. And that's just moving up through the cohesive forces, the hydrogen bonding within the water, and also the adhesive forces between the water molecules and the side of this xylem tube here, this cell here. So that's xylem, and water is moving up there, and dissolved in the water are ions. Now, these ones here, those are the phlegm, and moving through those is transport of substances that the plant has made itself. So things like sucrose that has been made from glucose subunits during photosynthesis. So that's transport of substances that the plant has made itself. And then out here, number 3 is the cortex. And the plant is using that to store starch. If you remember earlier on, we were talking about amylose, one of the polysaccharides in starch. Starch is large sort of complex in the plant's use to store as a store of energy, starch. Now, so you then have to look through the answers here. Well, number 1 is transporting water and ions. So number 1, transport ions. Well, it could be that one, couldn't it? Or that one there. Now the starch is in number 3. So store starch is correct for that one, and that one. And the transport of sugar is happening in our number 2, isn't it? There and there. And so the only row which has a tick in all columns is B. And the examiner was pretty blunt at that point and just said candidates didn't know their sections of vascular bundles in plants. You know, it's just really your understanding of transport in plants and being able to map that understanding of transporting plants to the structure and the function of the cells that are involved. Okay, number 26, we just did, we did that one, didn't we? So let's do number 27 is the next one. The examiner flagged up. So that was 25, the examiner flagged up, not 26, sorry. So 27 is now looking at oxygen dissociation curves. So this axis up here is telling us how strongly, it's telling us how much oxygen is still bound to a hemoglobin molecule. So here's your hemoglobin molecule, and you're binding oxygen to it. In human hemoglobin, you remember there's 4 heme groups, each that can bind to an oxygen molecule. So you might say, at least for human hemoglobin, hemoglobin plus 4 oxygen molecules is going to HbO8, all right? And then at that point, the hemoglobin is fully saturated with oxygen. You might say at that point, it's 100% saturated, but before any oxygen was added to it, it was 0% saturated. So this axis up here is telling us how much of the hemoglobin molecule is saturated with oxygen up to its maximum capacity. All right, and then this axis along the bottom is just telling us, well, you know, in what are the conditions surrounding the hemoglobin molecule where it starts to, or what are the prevailing conditions around the hemoglobin molecule? What's the, essentially the amount of oxygen that's surrounding it? And you had to choose between a human, a fish that's living in water where there's very low dissolved oxygen concentration, and a mammal that's extremely active and whose tissues have a very high rate of respiration. Well, I think the easiest one to spot to begin with is the fish. Now, the fish is in water, and that water has a very low dissolved oxygen concentration. So the fish needs hemoglobin that can grab oxygen when the concentration of oxygen is low. Now look at these. Now, which of those shows hemoglobin that can most strongly grab oxygen when the concentration or the partial pressure of oxygen is low? Well, the partial pressure of oxygen is low here, and that's another way of saying the dissolved oxygen concentration that the fish is in is low. And you can see here, out of all these, X, you know, even at low concentrations, can still saturate its hemoglobin to 80%. That's incredible. So that one must be the fish living in the water with the low dissolved oxygen concentration. It needs to breathe, and it has hemoglobin that can bind oxygen, even though the oxygen is at a very low partial pressure or low dissolved oxygen concentration in the water. So X is definitely this fish. Now, the next one that stands out is this mammal who is extremely active. And so in its cells, there's a very high rate of respiration. And that means that in the cells, well, a high rate of respiration will mean that the oxygen concentration, again, is going to be low because oxygen is being used up by the very high rate of respiration. So we want to see then a hemoglobin that releases oxygen when the concentration of oxygen is low, which is the opposite case to the fish, isn't it? And you can see here that Z must be this animal with a very high rate of respiration in its cells, because even at the low concentration, the hemoglobin is releasing oxygen. It's not binding it anymore. It's the reaction, this reversible process, is that at the low concentration of oxygen, the hemoglobin is not very saturated. It's only about 20 or 25% saturated, which means the hemoglobin is releasing its oxygen. It's going this way. So the oxygen is being released, and that's exactly what we want, because this mammal has very active respiration in its cells, and it's going to need oxygen to be part of that respiration that's happening in the cells. So Z then is the mammal with a very high rate of respiration. We've identified that. So Y is the only one left standing. That must be the human. So that must be then that number one. Sorry, X is the this again. Once you've figured everything out from the diagram, you've still got some work to do about plugging the right values into this table here, and it's that one there. Let's just check that. So number one is the adult human. Yes, we got that. Number two is the fish, isn't it? And that's graph X. There we go. The fish is able to get the oxygen from the low concentration in the water. And number three is Z, which is the mammal, which is the very high rate of respiration in its tissues. And that needs the hemoglobin the oxyhemoglobin to release its oxygen, so that the oxygen is then free to be used in the high rate of respiration inside the cells. Okay, so that was the correct answer for question 27. And the examiner said that the only students who could interpret this oxygen dissociation curve were really able to answer that. And you had to be able to interpret that curve both for the uptake of oxygen in the case of the fish and the release of oxygen from hemoglobin in the case of the animal. So you had to understand these dissociation curves both for the uptake of oxygen and the release of oxygen by the hemoglobin molecule. And possibly you normally come across those dissociation curves and you consider them one at a time. You either consider them when you're thinking about the uptake of oxygen, or you're thinking of them when you're looking at the release of oxygen. But this question is asking you to consider one set of dissociation curves for oxygen, both in those two different contexts, one in the uptake of oxygen and one in the release of oxygen. Okay, 29. This is a question about carbonic anhydrase, and really you're trying to understand there the reaction that takes place in a red blood cell, because carbonic anhydrase is one of the reactions, one of the enzymes that's present in the red blood cell. And what the examiner is trying to get you to work on here is that in the red blood cell, what you've got is the red blood cell has come from the lungs, and the hemoglobin is carrying oxygen, and that's coming through the capillary. And of course, what you want to happen is that the oxygen is released from the red blood cell. It has to somehow be released from its binding to hemoglobin into the cell where it can be used for respiration. Now that release of oxygen from hemoglobin is facilitated by hydrogen ions, and that process produces the hemoglobin without the oxygen molecule, and the oxygen molecule is released, ready to be transferred through the capillary into the cell for respiration. So to make that process happen, to make the hemoglobin release its oxygen, you need more hydrogen ions. And that's where this enzyme carbonic anhydrase is going to come into play, because carbonic anhydrase takes the carbon dioxide, which is plentiful from the cell respiration, and water, which of course is available as well, and carbonic anhydrase catalyzes that reaction to form, well, the hydrogen ion, which is the one that we need here to bind to the hemoglobin to release the oxygen, which is going to be used in the cell. And if you balance that equation, we've got the hydrogen ion, which has been donated from the water molecule, and that's going to be joining to the carbon dioxide molecule. And you've got this hydrogen carbonate anion there. All right. So the carbonic anhydrase is catalyzing that reaction to take carbon dioxide and water and make a hydrogen ion and the the hydrogen carbonate ion, and then the hydrogen ion that's being produced in that first step is then used to bind to the hemoglobin so that it reduces oxygen. So what happens after carbonic anhydrase has catalyzed a reaction? There it is, involved in the transport of carbon dioxide. Well, there's a dissociation of oxyhemoglobin, there it is, into hemoglobin, there it is, and oxygen. So that question A is the one that the examiner is wanting you to get there. And the examiner's comment on that again showed that many candidates didn't understand that you've got these two processes here, one that is catalyzed by the carbonic anhydrase, and the second one that uses a product of that catalysis, the hydrogen ion, and uses it to bind with the oxyhemoglobin to reduce the oxygen which is used in cell respiration. Okay, so what question was that? Was question 29. Next question that the examiner flagged up was this one here, which was a kind of, really, I mean, it's really about a sort of scientific method, really. So you've got here a scatter graph where they've plotted on the y-axis here the birth weight of babies, and then on the x-axis here the number of cigarettes smoked by the baby's mother each day during the pregnancy, and you get this kind of scatter here. And so you've got to try and sort of interpret that. And the first thing says, well, what conclusion can be drawn from this graph? Well, the first one, you have to look through each of these candidates. It says, well, the number of cigarettes, as the number of cigarettes increase, the weight of the baby, look at this, read this carefully, the baby's weight always decreases. Now, there is a correlation, isn't there? You'd have to say that generally it's true that there's a correlation between the number of cigarettes smoked each day. If that's greater, then generally there's a correlation that shows that the birth weight of the babies to those mothers is decreased. You can see that correlation. But it's not an exact correlation. There's some scatter here. Because, for example, if you take this point here and this point here, well, in the case of this baby, let's say the mother might have smoked 12 cigarettes a day, somewhere on that scale, whereas this mother smoked 20 cigarettes a day. And the one that smoked the greater number of cigarettes, in this case, had a higher birth weight than the mother of the baby whose mother smoked less cigarettes. So it's not true that as the number of cigarettes smokers, the number of cigarettes that get smoked increases, that the weight of the baby always decreases. Now, there's quite a lot of evidence to show there's a correlation, but it's not always true. There's an example here where one mother was smoking more cigarettes per day and had a baby whose birth weight was quite high. So that's not true. Now, so that must mean that there must be other factors that are affecting the weight of the baby, because you can see between these two that well, there must be some other factor that's affecting the weight of the baby, because even with 20 cigarettes per day, that baby up there had a relatively high birth weight. So it's true that there must be some other factors other than smoking that are affecting the weight of the baby. Now, there's a point here that we know that in cigarette smoke, there's all these various different constituents of cigarette smoke, and it's saying that nicotine, tar, and carbon monoxide slow the growth of the unborn baby. Well, what's happening there is you're proposing a causation, aren't you? Saying that these things, there's actually a reason that these constituents of cigarette smoke affect the birth weight of the baby. Now, this graph can show, I think, that there's a correlation. You can generally see that if we drew a kind of best fit line through there, then it's generally heading that way. There's quite a lot of scatter. So there are other factors that are affecting the weight of the baby. That's two, we had that already. So we can see there's a correlation, but the correlation does not imply a particular causation. Now we can come up with a hypothesis that actually maybe carbon monoxide is the causative feature for the low birth weight. Once you've got, you've made a hypothesis, then you're going to have to show there's an actual mechanism that ties the causation to the correlation. And only then can you say, well, in our case, just making that up, that the carbon monoxide actually is the reason for this correlation. Now, just because all we've got at the moment is a correlation, then you can't claim yet a causation. So that claim for a causation is not yet proved because we haven't proved the mechanism connecting the causation and the correlation. And this last one here, the majority of smokers smoke between 10 and 35 cigarettes per day. Well, that's an obvious distraction if you look carefully, because this was telling us about the number of cigarettes smoked each day by pregnant mothers. And this is telling us about the majority of all smokers. So that includes pregnant and non-pregnant mothers, women who aren't mothers, men, you know, all the people who smoke. We have no data about all the people who smoke at all. So that's clearly wrong. All right. So we've just been through these, and it looks as though the only one that is a conclusion that we can really draw from this graph is question C. There. And so the examiner said on this question here that only a few candidates were able to answer this carefully. So, well, I think, you know, we went through those in some detail, and it's true, if you work carefully through those, then you can see that actually number 2 is the only one that is a conclusion that can be drawn from the graph. The others you have to think about a little bit, but if you give them a bit of thought, especially this last one here, you can exclude pretty quickly. But the others, if you give them a bit of thought, you can exclude them as conclusions that you can really be drawn from the graph. Okay, so coming up then to the last question the examiner flagged up, which was 37, and that's talking about an understanding of the immune response. And this is an area that many students find difficult to remember, because actually the cascade of reactions and interactions and activations that happen when a pathogen enters the body and the body starts to recognize a non-self antigen, that whole cascade of what happens is a pretty complex cascade. And the examiner is saying that, well, you've got to learn that cascade, because at the end of that cascade, both B lymphocytes and T lymphocytes are both activated. And when they've been activated, then some of them are used in the immediate response to fighting the infection. But these activated cells can remain as memory cells. All right, so those two can remain as memory cells, and they are, both of those, they are part of the I spelled it wrong. So they are part of the specific immune response, aren't they? Whereas the phagocytes are part of the non-specific immune response. And so the memory of the specific non-self antigen that was dealt with in a particular immune response, well, you know, because it's a memory, it has to be a memory of something specific. So that excludes the phagocytes, such as the macrophages. And then you need to remember, or you need to work, or you need to, I don't know, draw some kind of sketch of the cascade, because you need to learn this cascade. and understand that both the B lymphocytes are activated and the T lymphocytes are activated. They are part of the specific immune response. And once these have been activated, then after the body has dealt with a particular pathogen and the antigen that it's presenting, then you can have these long-lived memory cells which have a memory of a specific response to one specific antigen. So both beta lymphocytes and T lymphocytes become memory cells. And the examiner is saying that many students said that only B lymphocytes become memory cells. I think that's probably easier to remember because these B lymphocytes are the ones that produce these specific antibodies, and so we think of those as being highly tied to having a long-lasting immune response. But both, if you look at the cascade, both B lymphocytes and T lymphocytes are activated. They're specific, and after activation, they can form long-lived memory cells of the immune response to a specific antigen. Okay, so we haven't been through each question. A lot of those questions you can look up, and then you can look up the answers either in the textbook or some of the ZNotes if you need to. But we have tried to focus on the particular questions the examiner flagged up that students had the most difficulty answering, and look at those in some more detail during this masterclass. And of course, if there's any further questions you have from this masterclass, you're very welcome to post your questions on the Slack channels, and the Z coaches on the Slack channels will be delighted to pick up your questions and help you build your understanding of AS level biology for Cambridge International Examinations. So thanks for joining this masterclass, and I wish you every success in your studies in biology.